If there's a biochemical message that you want to relay, one of the key ways is through an RTK. That is, a receptor tyrosine kinase, which, along with our G protein coupled receptors, our GPCRs, are two of the main ways that our bodies are able to transduce signals. So get a message from outside the cell, inside the cell, and let the cell respond. There are a lot of times when things are going on outside of the cell and you want to get that knowledge of what's going on outside, inside. This requires signal transduction, basically getting that message across to things that are in the cell. But a lot of times the message itself isn't going to come into the cell directly, like the messenger isn't going to come into the cell, but the message is still going to get relayed. And two of the main ways that this message can get relayed without the messenger, without the kind of like ligand having to actually come in, are G protein coupled receptors and receptor enzymes, so commonly receptor tyrosine kinases. Your kind of classical G protein coupled receptor, or abbreviated GPCR, is going to be your beta adrenergic receptor, and your classical RTK would be like your insulin receptor. More about insulin when we get deep into metabolism, but insulin is a peptide hormone. It's this small little protein that gets secreted by your pancreas, and it gets secreted when you have high blood glucose. So when your blood sugar is high, maybe you just ate a meal, your pancreas is going to secrete insulin, and this is going to tell cells to let glucose in. It's also going to tell them to link those glucose units up to form glycogen in a process called glycogenesis. The opposite of that is glycogenolysis, the breaking down of glycogen, which is going to be stimulated by kind of the opposite of insulin, which is glucagon, is another hormone that is produced by your pancreas. This time the alpha cells. And glucagon is actually going to signal through the same pathway as your beta adrenergic receptors. And insulin and glucagon are going to have opposing effects on glycogen metabolism. And so more on this later in the video a little bit. But for now, know that insulin is going to be kind of this hormone that's secreted when you have high blood glucose, when your cells need to take in the sugar, build that up into glycogen, and things like this. Insulin. Oh, and quick note, if you're wondering about diabetes, so people with type 1 diabetes, they don't make enough insulin, and people with type 2 diabetes, their cells aren't responding well to that insulin. More on that later, but for now, just know that it's this peptide hormone that is then able to bind to these receptors in the cells. This insulin receptor, this INSR, which is going to be the focus of our RTK talk. These proteins are embedded in a membrane, and on the outside, they have a ligand binding part that binds to some cellular messenger. And then that causes a conformational change. It causes a shape shift in the protein itself that then is able to relay the message on. Now, with our G protein coupled receptor, this is going to be through a G protein, and the receptor itself is not an enzyme. But that G protein is, and it can interact with enzymes that can help pass the message on, amplify the signal. In the case of our receptor tyrosine kinases, here, the receptor itself is an enzyme, and it's going to be able to directly relay that message on after getting activated. Its kinase activity gets activated by the ligand binding, and then it's able to pass the message along. And so phosphorylation is a great way to pass the message along because you can kind of put a little phosphate group on different molecules and target them and get them to interact, offer new like binding opportunities for other proteins. And so we'll see lots of scaffolding. We'll see lots of all those concepts about integrating signals, all that great stuff that comes with signal transduction that you can see examples of in both the GPCRs and the RTKs. In this video, we're going to be focusing on these RTKs, but there's also the video on GPCRs if you're interested. Although we're going to be focusing on these RTKs, both these and GPCRs are often going to involve G proteins. GPCRs, by definition, need to, but we'll see that one of the downstream players of our RTKs is going to be a G protein, this protein RAS. So we need to go step back and discuss what a G protein actually is. A G protein is a small little protein. It can be monomeric, so it can hang out by itself, like we see with the RAS protein in receptor tyrosine kinase pathways, or it can be heteromeric, so it can be part of, say, a G protein coupled receptor. The G alpha, that's going to be the part that actually binds to the GTP or the GDP, because what these enzymes do, yes, they're enzymes, is they actually hydrolyze GTP to give you GDP. Now, GTP, that's just like ATP, except it's got guanosine. Instead of adenosine, it's still got those 3 phosphate groups. And you can hydrolyze the last phosphate group off, just like you can hydrolyze the last phosphate group off of ATP. In this case, when this G protein is bound to GTP, we consider this the quote-unquote active form of the G protein. Here, the activity is not referring to enzymatic activity, but rather the form of the G protein that's in the kind of like shape that's able to interact with partners and kind of activate the partners. This happens because this last phosphate group, this gamma phosphate, the one that's actually going to get hydrolyzed off, remember when we say hydrolyze, that's basically just use water to cut off. That's actually going to be making interactions with these regions of the protein. So when you cut that off, well, now you kind of like have the protein change shape, and that's going to make it so it no longer interacts with those interaction partners. You can think of it kind of like kicking off their partners. And when it kicks off their partners, well, then those partners aren't getting that stimulus anymore. And this protein is then going to be bound to GDP. So the G protein has this activity where it is able to hydrolyze the GTP to take itself to the GDP form. If it was just constantly just inactivating itself, that wouldn't be very useful. And so this hydrolyzed activity is actually pretty darn weak. And it needs help to actually convince it to do that hydrolysis. And so to convince it to do that hydrolysis, you have GAPs, which are GTPase activator proteins, which are going to kind of stimulate that cleavage, facilitate the hydrolysis. And then this is going to inactivate the G protein. So you have these GAPs that are going to activate the enzymatic activity, which will deactivate the protein. So when the G protein does what it does as an enzyme, it inactivates itself. So it breaks the GTP, and now it's bound with to GDP. Now, it needs to actually swap out that GDP for a new GTP. It can't make GTP. It can only break GTP. And it turns out that it holds tighter to that GDP. So once it inactivates itself and now is holding onto this GDP, the GDP is going to be bound in there pretty tight, and it's going to have to get kind of kicked out. And so the GDP got to get pushed out with the help of a GEF, or GTP-GDP exchange factor. And so the GTP-GDP exchange factor, these are going to help them exchange that GDP that they're bound to for a new GTP. So once you kind of remove the GDP, well, it turns out the concentration of GTP is high enough that it can kind of pick up a new GTP pretty quickly, as long as the GDP wasn't just there hogging things. And that G protein, now that it's bound to the GTP, it's now active and it can go activate other things. So to review, the G protein is going to be an enzyme that hydrolyzes GTP to give you GDP. It hydrolyzes GTP, but it's not going to go and make GTP. Just like it takes energy to make ATP, which is why we have to do all of this like oxidative phosphorylation stuff and use a bunch of energy that we get from breaking down molecules. Well, it's also going to require energy to make GTP. And in fact, you can also make GTP from ATP that you made in those other pathways. So you got a lot of GTP hanging around. Therefore, when you hydrolyze that GTP, and then you get GDP, and you kick out the GDP, now a new GTP combines. These G proteins are pretty slow naturally, and so you need help in both the hydrolysis, which is helped by a GAP, and in the exchange of the GDP for GTP, which is helped out by the GAP. And so you see various GAPs and various GEFs that can interact with these G proteins and help modulate their activity so that these proteins are able to kind of act as these switches and turn on and off entire pathways. Pretty cool. What's not so cool is that they are often mutated in various cancers, because they have such a crucial role, if you mutate them, you can permanently kind of activate pathways, and then you can if these pathways are involved in cell growth, then you can develop cancer because, well, cancer is just uncontrolled cell growth. We can also find G proteins in other places in biochemistry, such as in the process of translation or the making of proteins. We have a variety of G proteins that are actually going to be helping kind of move things along in the ribosome. In the case of our G protein coupled receptor, remember what our receptor was? It was that 7 transmembrane protein that was going to not be an enzyme itself, but it was attached to an enzyme. It was attached to that G protein that could ultimately act as a GTPase and then activate itself, act as that switch. And the role of the receptor was actually as one of those guanosine exchange factors to get that G protein to dump its GDP and form and pick up a GTP, activate that, and cause the signaling. In the case of our receptor tyrosine kinase, here our receptor is itself an enzyme. It's going to be a tyrosine kinase. When we talk about kinases, basically the name before the kinase, the amino acid before the kinase is telling you what it phosphorylates, and so it phosphorylates tyrosines. More specifically, these receptor tyrosine kinases, they can actually phosphorylate themselves, and they can phosphorylate other things as well. But in order to be activated, they have to first phosphorylate themselves. And when something's phosphorylating itself, we call it autophosphorylation. So it actually phosphorylates like the other copy of itself. Receptor tyrosine kinases are going to act in like dimers. And so sometimes we'll see that they're going to be constitutive dimers, such as in the case of our insulin receptor, or INSR. It's going to act as a constitutive dimer. So it's always a dimer, but it's not active. On the other hand, we have other sorts of receptor tyrosine kinases that are going to be monomeric, and then only upon the binding of a ligand do they come together. So with our insulin receptor, basically here, the binding of a ligand activates it, but it doesn't cause the dimerization. It's already a dimer. But with these other ones, binding of a ligand causes the dimerization and then the activation. Once you have the activation of one of these, basically they autophosphorylate one another, then they start phosphorylating other proteins, leading to a kinase signaling cascade. How do you actually activate these? In the case of the insulin receptor, you have this dimer. So remember, it's this constitutive dimer, but it's going to have an inactive kinase domains. These tyrosine kinase domains, they're going to be capable of phosphorylating tyrosines, including tyrosines on themselves or on the other kind of like monomer, the other copy of itself. But they can't do this unless they kind of have the right conformation. And they won't have the right conformation unless you actually have the binding of the ligand. So the binding of the ligand causes a conformational change that's going to activate the subunits to activate one another. So basically it changes the shape of these subunits so that they're then able to go and phosphorylate one another. They basically phosphorylate this key part called the activation loop. This activation loop was blocking the substrate binding site. So the part where kind of like the active site where the substrate needs to bind to get phosphorylated, you have this activation loop that's blocking it. If you phosphorylate the activation loop, well, now you move that loop out of the way and allow substrate to bind. These monomers can basically phosphorylate one another's activation loops to move those loops out of the way and now cause the substrate binding site to be open so that they can phosphorylate things. It can then go and it can phosphorylate this protein called IRS1. Now, IRS1, once it's phosphorylated, it's going to serve as sort of this kind of connection point. So IRS1 stands for insulin receptor substrate 1. And what it does is it's kind of just this hub. So it has these sites that get phosphorylated by the insulin receptor, but only remember if the insulin receptor is activated. So if the insulin receptor binds to insulin, it activates itself, which is then going to go and phosphorylate IRS1. Once IRS1 is phosphorylated, well, now it's going to have a binding site for a protein called GRB2. And GRB2 is basically then going to bind to this protein called SOS. SOS is then going to bind to this protein called RAS. And what's RAS? Well, RAS is one of our G proteins. And so RAS is one of those little G proteins that's basically, this one's monomeric, but it's able to do just what all G proteins do. It's able to take GTP and break it down. And it's able to then exchange that GDP, the end product of that breakdown, the GDP, it can dump that and bind to GTP. But remember, that's going to be a slow process. And so we can have proteins that are going to speed things up. And one of the proteins that can speed things up is going to be this SOS protein. This SOS protein is then therefore going to act as one of those guanosine exchange factors. It's going to help RAS switch out the GDP for the GTP and activate RAS. The activated RAS, well, it is going to bind and activate RAF1. Now, RAF is going to be a kinase, and it's going to set off a signaling cascade of kinases. And we have one kinase, and it can phosphorylate a lot of stuff. What if we have a kinase phosphorylated kinase? Well, now we can have that one kinase phosphorylate a lot of other kinases, which can phosphorylate a lot of other kinases, which can phosphorylate another kinases, which can phosphorylate a lot of other kinases. And so in this way, we're going to get a big signal amplification, even without the production of any really like secondary messenger. We can have this phosphorylation cascade where ultimately we can phosphorylate a protein that can then go into the nucleus and affect the transcription of genes. And so this is one of the common pathways that we'll see is this like MEK pathway. So we had ultimately this protein ERK that was the one that was actually getting into the nucleus. And it turns out that ERK, as well as MEK, so the thing that activated ERK, as well as RAF, which was the thing that activated MEK, these are all basically in larger families. And these families have kind of ridiculous names. ERK is in the MAPK family for myosin-activated protein kinases. And it's specific for serine or threonine residues. Instead of phosphorylating tyrosines, it phosphorylates serine or threonine residues. And not just any ones, it has like all kinases, they have like consensus sequences, which are the things that they like to phosphorylate. So they're not just phosphorylating anything. But it phosphorylates specific serine or threonine residues. And it can phosphorylate things like transcription factors, such as ALK1, which is then going to activate them and cause differences in the transcription of different genes. And often for like insulin, you'd have things that were needed for cell division. So if we had ERK be in this MAPK family, well, it was activated by MEK. And MEK is in the MAP kinase family. So basically, it's a kinase that activates the MAP kinase. And so MEK is going to be in the MAPKK family. And then if we had a MAP kinase that was activated by another kinase, well, that other kinase would be a MAP kinase. So RAF1 is in the MAP kinase or MAPKKK family. And so basically, yeah, the naming can get complicated. What I find is the most useful is just kind of being able to look at pathways and look at arrows, what's going where, and not try to memorize everything. Speaking of what's going where, how do things know like where to go? Basically, if you imagine how many different proteins are in a cell, you're going to need a way to kind of hold things together. And one of the ways that you'll do this is with these scaffolding proteins. So this is one example, this kinase suppressor of RAS. Basically, this is a scaffolding protein that's kind of going to hold together all those components of the pathway so that things can find each other more easily. How do these things all kind of like bind together? A lot of it is going to be mediated by these, by binding to phosphorylation sites. So there are a couple of main kind of like phosphate binding domains. So one is this SH2 domain, which is a protein domain that binds to phosphorylated tyrosine residues. We also have SH3 and pH domains, which are protein domains that bind the membrane phospholipid PIP3. So we have phosphorylation-mediated ways in which we could kind of bind to membranes and which we could bind to phosphorylated proteins. And what you see a lot is that basically different types of proteins are going to have different combinations of these SH2 and SH3 domains, as well as a lot of other domains. By kind of like having all of these different domains and all of these sites, you're then able to build these kind of places where these proteins can interact with one another in specific ways that are going to be mediated by phosphorylation. So though we talk about like the SH2 domain as being like binding phosphorylated sites, remember that it's not only just that one phosphosite that's going to be important for distinguishing between different proteins, there's going to be a lot of interactions. But by having this one like place where it can kind of recognize phosphorylated versus not phosphorylated, you can therefore regulate when things are bound where. So you bring things together, say only when they're in the phosphorylated or the not phosphorylated form. Remember we said RAS is one of those G proteins. So in this signaling pathway of insulin signaling through RAS, we are using a G protein, but we're not using a G protein coupled receptor. Our G protein is not directly attached to our receptor, and it's monomeric instead of being heterotrimeric. So you can see that basically RAS is down the line from our receptor, connected through the IRS1, which is bound to GREB2, and which was bound to SOS, which was going to be bound to our RAS. Now, RAS you might have heard about because it's going to be very important in the context of cancer. Mutations in RAS that like eliminate its GTP as activity are going to cause it to remain constitutively active or constantly active. These sorts of mutation occur in about 25% of human cancers. You can also get mutations in gaps. So those proteins that were actually going to cause the G protein to activate its GTPase activity. So basically the mutation in RAS itself can alter the natural GTPase, like the intrinsic GTPase activity of the G protein. But you can also have mutations in the regulatory proteins that regulate the G proteins, such as this tumor suppressor gene, NF1. So NF1 encodes a gap, which causes RAS to stay active for an extended period if you have a mutation that's going to inactivate that NF1. When we talk about a tumor suppressor gene, basically that's a gene whose normal function is to kind of like keep a check on cell growth. If you have a mutation in one of those, well, now you're going to have, you're removing the brakes. There are also proteins that we can call like oncogenes or proto-oncogenes. If the normal gene would be a proto-oncogene, and then when it's mutated, it becomes an oncogene. And these sorts of genes are things that are going to kind of stimulate growth, division, this sorts of thing. So that was one pathway that we could go with the insulin receptor, where we had this adaptive proteins, the RAS activating protein, the RAS, the MAP kinase, the MAP kinase, the MAP kinase, and then into this nucleus and affecting things. But we also have tyrosine kinase receptors that are able to actually act through a different pathway. And so they can actually act through that same phospholipase C pathway that we saw before. Additionally, insulin can act through this PI3K pathway. I like to think of insulin as saying, better let some glucose in, because they're produced by beta cells of the pancreas, and it tells the cells, hey, let's let in some glucose, the blood sugar is high, let's let this glucose in, and let's link it up to form glycogen. So let's take those individual glucose units and let's link them up, and this is a process called glycogenesis. On the opposite hand of things, you have this hormone glucagon. Glucagon says all the glucose is gone. It's made by the alpha cells of your pancreas, and it does the opposite of glucagon. It's going to tell the cells to break down that glycogen. The liver is going to ship out sugar for the body. The muscles are going to use that glycogen for energy. And so one of the signals that's going to tell them to do that glycogenolysis, that breakdown of glycogen, is going to be glucagon, but another signal is also going to be your epinephrine, or remember that's the other word for adrenaline. Both adrenaline and glucagon are able to act through the same signaling pathway involving the production of cyclic AMP, the activation of PKA, and then ultimately the breakdown of glycogen. Because it's that GS alpha part that actually, that's the G protein that actually interacts with the cyclic AMP, they can have different subunits of the other parts that have different kind of adapters to respond to glucagon or to adrenaline. Long story short, the insulin pathway and the glucagon pathway are actually going to be opposing one another. Same with the insulin pathway and the adrenaline pathway, because remember, those are going to be the same. And so when we talk about the beta adrenergic receptors, also think in your head the glucagon pathway. Glucagon, however, is only going to be in only certain cells are going to respond, like your hepatocytes in your liver, which is one of the places you store glycogen. So that's able to break it down and chip it out. More on this when we talk about metabolism, but for now, just know that insulin and glucagon are going to have these opposite effects when it comes to glycogenesis. So the making of glycogen, which is stimulated by insulin, and glycogenolysis, the breakdown of glycogen, which is stimulated by glucagon or epinephrine. They're going to be acting on this same pathway with opposing results. So one of them will be like dephosphorylating, where the other's phosphorylating, and things like this to regulate this breakdown or the making. Remember that IRS1 was getting phosphorylated by our insulin receptor. Here, once phosphorylated, it associated with this GRB2 kind of like scaffolding protein, which is associated with SOS, which activated RAS. In the case of this other pathway, here what's going to happen is that phosphorylated IRS1 is going to interact with this PI3K. So IRS1, once it's phosphorylated, well, now it can bind to PI3K. When you see a K, think kinase. This is going to be a kinase. It has an SH2 domain that's able to bind to the phosphorylated IRS1 and get activated. Once activated, well, here it can basically phosphorylate PIP2. PIP2 is phosphatidylinositol 4,5-bisphosphate, which is one of our phospholipids. PI3K is basically able to phosphorylate that to give you PIP3. Don't confuse this with IP3. This is still like a full phospholipid, but it has a third phosphate group on this phosphatidylinositol head. And once it's phosphorylated, well, now it can bind to PKB. Another K, another kinase, we've got protein kinase B. Protein kinase B is going to bind to PIP3, and it's going to be phosphorylated by another kinase, which is called PDK1. And then this is going to be fully activated. Now, this PKB is going to phosphorylate a protein called GSK3. Why do we care about GSK3? Well, GSK3's normal role is to regulate this glycogen synthase. And so glycogen synthase making glycogen, remember that's our storage unit for glucose. So it can build glycogen from glucose and help you store glucose. So in the response to epinephrine, remember we said we wanted to kind of like break down glycogen. In the response to insulin, we're going to want to build up glycogen. So what we're going to want to do is we're going to want to have our glycogen synthase be active. Glycogen synthase is going to be regulated. Its activity is regulated by this kinase called GSK3. GSK3, when it's active, it's going to phosphorylate glycogen synthase. So I know that a lot of times we talk about phosphorylation as activating things, but in this case, phosphorylation is actually going to inactivate our glycogen synthase. Once it's inactive, it can't make glycogen. If we have GSK3 active, well, our glycogen synthase is going to be inactive. But if we inactivate GSK3, well, now we prevent it from inactivating GS, which means we can make glycogen. So PKB, what it's going to do is it's going to inactivate GSK3, and therefore it's going to turn the brakes off of the brakes on the glycogen maker. So it's like a complicated pathway, but at the end result, you get glycogen made. Also, what happens is that PKB, remember, it's a kinase, it can phosphorylate. It doesn't just have to phosphorylate a single thing. It's an enzyme. It can phosphorylate over and over and over again, and it can phosphorylate different molecules. And so each kinase is going to have like specific molecules that'll phosphorylate. They have to have like that consensus sequence and everything, but they can still be multiple proteins that they're going to phosphorylate. In the case of PKB, one of the proteins that it's going to be able to act through are going to be other G proteins. You can't escape those G proteins in signaling. In this case, we have RAC1, RC1, and RAB. Long story short, what they're going to do is they're going to cause the movement of these these glucose channels from these like internal membranes to the surface of the cell, allowing glucose to be taken in. If you think about it, if you want to be able to take glucose into, like regulate how glucose gets into your cells, one way is by regulating how much of the glucose receptors and transporters are on the surface of the cell. But actually going and making those proteins and those transporters is going to take time. If you want to be able to respond more quickly, you're going to want to have them pre-made. You can kind of like have them pre-made but kind of like held inside the cell in these little kind of membrane pouches. And these internal vesicles can then merge with the membrane once they get the signal, and the signal is going to be relayed through these G proteins, allowing you to move those transporters to the surface and let glucose in. We saw how insulin could lead to these different pathways, and we can also have lots of different receptor tyrosine kinases. So again, insulin's kind of weird, or insulin receptor is kind of weird in that it's kind of a constitutive dimer, but most of these are going to be monomers usually. Some examples are these growth factor receptors, EGFR, various receptors. A lot of these are important for controlling cell division. So it's really important that cells tightly control their cell division, or if not, basically you get uncontrolled cell growth, which is cancer. So there's different phases to cell division, and basically there are different checkpoints at these different stages so that cells only divide when the time's right. But if there are problems with that, then you can get missignaling, you can get uncontrolled growth. One way in which cells kind of control things is through phosphorylation via cyclin-dependent protein kinases, or CDKs, which are made in response to growth factor-based signaling. So you have these growth factors that are going to lead through receptor tyrosine kinases to one of those MAPK cascades. They cause things like the phosphorylation of these proteins Jun and Phos in the nucleus, which are then going to activate this transcription factor, which are going to regulate the making of those cyclins and those CDKs, enzymes for DNA synthesis, and cause the cells to go from G1 to this S phase. So basically here in this S phase, they're making that DNA, they're preparing to divide. These signaling factors are going to help tell it to divide. And you can imagine that you could have mutations in all sorts of different places that could therefore cause this to happen. There are all sorts of different types of receptor tyrosine kinases, and they'll have different substrate specificities. So they'll bind to different types of primary messengers. Some of them actually don't have a known ligand, or some of them we don't think they actually have a ligand at all. And so there's this one called HER2. You might have heard about it in the context of breast cancer. HER2 is one of those ones that doesn't need a ligand to activate it, but it does need to form a dimer. And so normally you don't have a lot of HER2. And so you're not forming any of these many homodimers. But what happens, so those homodimers, they wouldn't need a ligand to be activated. But what could happen is basically often this HER2 is going to team up with another RTK, which did need a ligand. So you still had this ligand activation. In the case of overexpression of HER2, however, so if cells are making a lot of it, such as cancer cells that overexpress HER2, well, now they're able to make a lot of this functional dimer that doesn't need a ligand to be activated. If it doesn't need a ligand to be activated, well, now it's going to be constitutively active if there's enough of it where you form this dimers. One of the common mutations associated with breast cancer is an overexpression of HER2. And there are antibody treatments like Herceptin that actually go and try to block this receptor so that it can't be activated. You can't get that activity. There's also a lot of crosstalk between the pathways. So that insulin receptor, it can also go and it can phosphorylate the beta adrenergic receptor. IRS1, that was acting through PKB, which could phosphorylate the beta adrenergic receptor. All those things that are basically able to kind of control the internalization of those receptors. But you can also have it's basically the insulin receptor can phosphorylate a different site on the GPCR, which can then lead it to kind of be a building off point for one of those MAP cascades. So lots of crosstalk between the pathways, which a lot of it depending on kind of what's hooked up where. Both these receptor tyrosine kinases and these G protein coupled receptors and other types of receptors all can kind of take advantage of these similar strategies. You have crosstalk between the different pathways. You have similar but different results. All these cool things. Your cells are pretty darn smart, especially with the use of things like scaffolds to hold proteins together that you want interacting in this pathway. You're able to quickly transduce the message from outside, so relay that signal from outside the cell into the cell, and then also stop that signal quickly as well. Broad overview of signal transduction. We have those GPCRs where we have the G protein coupled receptors bound to a heterotrimeric G protein. The G alpha subunit is the one that's going to bind to GDP and exchange it for GTP upon response to the primary messenger, the signal relayed through the receptor. So the primary messenger, the hormone, is going to bind to the receptor, cause the receptor to change shape. The changed shaped receptor is then going to cause the G protein to change shape, to release its GDP and take up GTP. Once bound to GTP, well, now it kind of exposes a binding site for adenylyl cyclase. And so it's going to ditch that those beta and the gamma subunits, go and find the adenylyl cyclase and activate it. Adenylyl cyclase is then able to produce cyclic AMP, our second messenger, which can then go activate things like protein kinase A, which can activate other things. Alternatively, it can go in and can activate protein lipase C, which is basically going to lead to the production of IP3 and DAG, which are then going to act through a different pathway. We can also have our receptor tyrosine kinases. Here, these are going to function as dimers. The ligand binds, activates autophosphorylation. Those dimers are going to phosphorylate one another and lead to signaling cascades through kinases typically, as well as pathways that can act through other lipid-mediated functions. We also have our gated ion channels where things are going to open up in response to a stimulus. And we have things like steroids, which can just go directly into the nucleus and bind to receptors there to alter transcription. There's still a lot more that I didn't go into, but